Hello everyone. This presentation is about disease control. This is also made by a student like you. So disease control, prophylactic measure, curative measure and biological measure. This presentation is prepared by Patel Anjali. Introduction. So plant disease control is to prevent qualitative and economic losses of crops due to attack by pathogens. Various methods adopted for control of plant diseases may be classified into the following three categories: prophylactic measures, curative measures, biological measures. So, prophylactic measures, they are preventive and include protecting the host from exposure to the pathogen, from infection, or from environmental factors favorable to disease development. Some important prophylactic measures in operation are given below: quarantine, eradication alternate host, crop rotation, sanitation, environmental manipulation, chemical prophylactic. Quarantine, it is the legal restriction at the national or international level on the trade of propagation. This is done to prevent the spread of disease. The first Quarantine Act was enforced in 1912 in USA. In India, Destructive Insects and Pests Act was passed by the government in 1914, and it has been amended from time to time. Thus, quarantine regulates entry of the plant material in a very effective way. However, several diseases have been introduced in India even after the enforcement of DIP Act. These include leaf rust of coffee, lead blight of potato, and boll disease of potato. So, eradication of alternate host. The alternate host usually provides shelter to the pathogen during unfavorable conditions. Thus, eradication of collateral host is a good measure for the control of heterogeneous pathogen. Barberry eradication in temperate countries help in the control of black rust of wheat caused by Puccinia graminis. Crop rotation, growing a particular crop or similar crop in the same field for several years continuously increase the soil-borne pathogen in the field, and the disease appears with increasing severity after every year. So ultimately, the soil becomes so heavily infested with the pathogen that it can no longer support the growth of the crop. Crop rotation helps in the control of such soil-borne diseases. Rotation of various crops reduces survival rate of the pathogen in the absence of the suitable host. Example, some important disease that can be controlled by crop rotation, like wilt of arahar, wilt of linseed, wheat burnt, rice, etc. Sanitation, it is the simplest method of disease control. It reduces the amount of inoculum to which the host populations is exposed. Several types of pathogens are present in various parts of plant in the soil, which act as inoculum for disease caused by them. Several infectious diseases like powdery mildew of wheat and peas, downy mildew of maize and peas, wilt of cotton can be controlled by such. In greenhouse, soil-borne disease can be controlled by the use of sterilized soil. Environmental manipulation, it includes such cultural practices which discourage pathogen development. Good soil drainage, proper aeration and irrigation, proper fertilization or soil amendment, proper spacing of plants or weed control are some important factors that discourage growth of the pathogen. Only disease-free certified seeds should be sown to achieve the purpose. Chemical prophylaxis. It is the application of chemicals to plant for control of pathogens. Chemicals used in plant protection are known as pesticides, and they include fungicides, bactericides, insecticides, and herbicides. Most of the pesticides are used to control disease of the foliage and other above group part of the plants, but some are used to disinfest and protect seeds, tubers, and bulbs from infection. There are two categories of fungicides: contact fungicides, residual fungicides. Contact. These fungicides act by direct contact with the pathogenic fungus before or after the infection. Residual fungicides. These fungicides are used before the infection of the pathogenic fungus. Hence, they form a protective covering over the surface of the plant even before infection. On the basis of their chemical nature, fungicides can be divided into two major groups: inorganic compounds and organic compounds. Inorganic compounds: Boldex mixture, it is specific against downy mildew, blights, white rust disease, etc. Burgundy mixture, it is comparatively less phytotoxic, hence it can be used safely on tender leaf plants. These are specifically used for control of leaf blight and related diseases. Chestnut compound, it is used in the control of damping of mercuric and mercurous chloride, used to control several surface fungal diseases. Sulfur sprays control powdery mildew of various crops and leaf curl of peach. Organic compounds, they are most versatile and widely used group of modern fungicides. Thiron, used in the control of disease caused by Pythium, Fusarium, Protomyces. Furban, used as protectant fungicide against a variety of fungal disease of fruits and vegetables. Example, early blight and late blight of potato. Manet, it is used as foliar spray against foliage and fruit disease of several vegetables. Zenet, it is safe and multi-purpose foliar and soil fungicide for early and late blight of potato and blast and leaf blight of rice. Zeram, it is effective against several foliar diseases of field and orchard crops. Nebem, it is effective against leaf blight and seedling of blights. Quinones, quinine, it is an important quinone mainly used for the seed, bulb, and flower treatment. Diclone is used and as protectant of vegetable seeds and as a protectant spray against some blights, fruit rots, and cankers of fruits and vegetables. Heterocyclic nitrogen compound, they are used as foliage protectants on the fruit and vegetable crops. Captain, effective against leaf spot, blight rots, etc. Fulbate, effective against powdery mildews. Difoltan, effective against early and late blight of potato. Benzene compounds, dinitrocresol, used as a spray for some disease of fruits and ornamental trees. Chloronab, effectively used to combat seedling disease of cotton. Terrachlor, effective against various soil-borne disease of vegetables and is primarily used against Rhizoctonia, Plasmodiophora. Systemic fungicides, these are usually absorbed by roots and are translocated within the plant in the transpiration stream and the latter thus become fungitoxic. They can also be injected into the tree trunk and reaches to different plant parts through the xylem stream. The inactive enzymes and toxins of the pathogen inhibit growth of the fungus. So there are three categories of systemic fungicides: oxaltins, benzimidazole, and pyrimidines. So antibiotic. Several antibiotics are also useful in the control of plant disease. They are generally absorbed and translocate systemically and act directly on the pathogen. They can be applied in the form of spray or dust. Example, streptomycin. It is commonly used against several pathogenic bacteria like Pseudomonas and Phytophthora. Other antibiotic, tetracycline, controlling the diseases caused by mycoplasma. Curative measure. Besides prophylactic measure, sometimes infected plants are individually cared by treatment. Such methods are known as curative or therapeutic measures. These measures are used after the appearance of diseased plant is necessary. For the protection of healthy plants, eradication of diseased plant is necessary. So these measures are effective against smut and blight diseases. The disease can be cured by physical or chemical therapy. The physical therapy includes surgery, temperature modification, radiation, and moisture. The chemotherapies are usually directly on the surface lesions or as solutions to be absorbed through roots. Bromide, Beta-Vax, plant wax, and dithin. These are the good chemothermophytants. Biological measures. The biological measures for disease control can be divided into the following three categories: breeding resistant variety, hyperparasitism, control through trap crops and antagonistic plants. Breeding resistant varieties, use of varieties which show resistance to infection of pathogenic organisms is an ideal measure for the control of plant disease. This is also known as illness method of disease control. High-yielding resistant varieties have been developed by breeding techniques. Some economically important varieties of wheat which are resistant to disease are as follows: rust-resistant varieties of wheat, Lerma rosa, Kalyansona, etc. Smut-resistant varieties of wheat, NP4 and NP710, etc. Hyperparasitism. Hyperparasitism is the control of pathogenic microbes with the help of other microbes which parasitize or antagonize the pathogen. Bacteriophages, mycophages, and nematophages. These are some fungi. These are some common examples of hyperparasitism. Several pathogenic fungi such as Pythium, Fusarium, and Helminthosporium are infected by other fungi present in the soil. So control through trap crops and antagonistic plants. There are several plants which are infected by fungi and nematodes, but after infection, the most secretes certain substances. They secrete certain substances which are inhibitory to these pathogens. Such hosts are known as crops. For example, populations of Heterodora nematodes can be reduced in the soil by Solanum nigrum and Crotalaria species. Trap the larvae of Melodogni. The population of several pathogenic microorganisms can be reduced in this way by rotation of trap crops. Thank you very much. If you like this presentation, please like, share, and subscribe. Hello everyone.